Hello, grade 12s. So we are now at part 4 of my series on meiosis. So I've talked about all the important things relating to meiosis, the structures, the terminology, the phases, some of the more SAGS related requirements that you need to know. But now we're going to move on to the more applied questions relating to meiosis. How do we use meiosis in the outside world a little bit more, as opposed to knowing that this is how we create gametes? So what are the specific things that help us to understand both disorders and how to understand those disorders? So talking of disorders, the first thing I'm going to be talking about is meiosis disorders. So I just want to take you back a little bit to when we talked about DNA. And remember, I talked mainly that there was 2 different types of mutations that you can have. The one being point mutations, so your substitutions, your insertions, your deletions, and the other one I said was chromosomal mutations, where the whole chromosome was affected at once. And I said that we were going to deal with these in meiosis. Well, that's what we're going to be dealing with now. We are dealing with chromosomal mutations, where a whole chromosome is additional or taken out at the same time. So I'm not dealing with point mutations, chromosomal mutations. So the main meiosis disorder category that you need to know about is aneuploidy, or chromosomal non-disjunction, that we like to talk about. Okay, so I know that that's a lot of big words, but let's basically break it down a little bit. What do I mean by aneuploidy? So it sounds kind of similar to a previous word that you maybe have encountered from the plant reproduction section, being polyploidy. So polyploidy refers to more than 2 sets of chromosomes. So remember, we as a normal organism have diploid chromosome number, 2 sets of each chromosome. But in a plant, for example, it is possible for you to be triploid, tetraploid, heptaploid, octoploid, up to 8 sets. A strawberry, the normal strawberries that you eat, are octoploid. They have 8 sets of chromosomes per chromosome, that whatever their number is. So that is normal for that plant. However, aneuploidy is when it is an abnormal chromosome number for that particular organism. So for us as a human, for example, we can't be triploid. We can't have 3 sets of the same chromosome or 3 sets of chromosomes in general. That is not how we function. In the same way, we cannot be haploid. So aneuploidy is when you get an abnormal chromosome number for what you normally are supposed to have. Chromosomal non-disjunction, what do I mean by that? Non-disjunction refers to there being an incorrect separation. So either an incorrect homologous chromosome separation or an incorrect sister chromatid separation. So let me show you what that means. This is an example of non-disjunction in chromosomes. So it's 2 different versions. Let's deal with the left-hand side here first. So in this left-hand side, the non-disjunction has occurred in meiosis 1. So in metaphase 1, I have the homologous chromosomes lining up in the middle. So in this cell, for example, there are 2 chromosomes. Centromere's number is 2. But because they have incorrectly separated, both of them have gone to the same side in this cell, meaning that this cell has 2 and this cell has none of this particular chromosome, where it should be 1 and 1. What then further happens in meiosis 2, that occurs normally, and what I should be having is 1 line, 1 line, 1 line, 1 line. But what I get is 0, 0, 2, and 2. So now what would happen if this gamete, for example, fused with the normal gamete of, so to say this is a human cell and this represented 1 pairing of chromosomes, if this gamete fused with a normal gamete of the other sex, I would be 1 chromosome short. Same with this one. Imagining that there's supposed to be 23 of these occurring, and the non-disjunction only occurred at 1 homologous pair. If one of these gametes fused with the opposite sex, I would have 1 extra chromosome for this one. I know that, go through that again for yourself. So imagine I have a normal set of 23 that are lining up on this side. along down the middle. Only one of the homologous pairs has a non-disjunction, and that only one of them is where I've got 2. All the others are just 1, 1, 1, 1, 1, 1. It would mean that eventually, for that homologous pair, I have none of them in gamete A and B, which would mean that if I fuse this with the other gamete, I then have 1 chromosome less, because I'm supposed to get this 1 chromosome. So I would then have 45 chromosomes instead of 46. If I have any of these where I've got 2 of the same chromosome, imagine all the other 22, I just have 1. So essentially this haploid cell has 24. If I fused that with a normal 23, I would then have 47 chromosomes. So these would only have 22 in them. So just get that into your head. Just pause, rewind, go through that again if you need to. So that's if the non-disjunction happens in meiosis 1. The non-disjunction can also happen on this side at meiosis 2, where the sister chromatids don't separate properly. So in this cell, there was a normal division. So I had 2 to start with. What am I finishing off as? With 1. Now in this cell, the non-disjunction has occurred. I start with 1, but I've got 0 and 2. But the sister chromatids are here. This is still 2 sister chromatids, 1 chromosome. Remember, eventually from there. So I still have an extra chromatid in this particular cell. So that is non-disjunction. This can happen at any of the chromosome numbers technically. So there's 23 different versions of this basically that you can have. But there's only 2 that you need to know from memory for aneuploidies non-disjunctions. The first is Down syndrome. So Down syndrome is characterized as having an extra chromosome 21. So at 21, you will have 3 chromosome 21s instead of having 2. Not that you have 1 less, it is 1 extra chromosome 21 is what leads to Down syndrome. So I know Down syndrome is probably one of the more common of the meiosis disorders that people have heard about. Things like Lifetime shows have sort of brought people with Down syndrome to the forefront so that people know about it. But there are obviously several physical symptoms that can occur because you have this extra chromosome 21, such as slanted eyes, small nose, abnormal ears, etc. But we also know that there is additional mental and social development issues that occur with Down syndrome as well. So there may be different forms or rates or amounts of mental retardation that occurs. Some may be more highly functioning Down syndrome people as opposed to less functioning. So there is a spectrum of Down syndrome effect that actually does occur. You don't need to worry about that, but just be aware of it. So you need to know that extra chromosome 21 is Down syndrome, and you need to know the characteristics of someone with Down syndrome from memory. The other one that you have to know about is Klinefelter syndrome. So in this instance, you have an extra X chromosome, and the sex chromosome combination will therefore be XXY. So what it's saying is you are a male XY with an extra chromosome X. If you were XXX, that is a completely different disorder. So XXY is Klinefelter syndrome. So essentially it's a male with an extra X chromosome. So what are some of the characteristics of someone with Klinefelter syndrome? Well, they will generally present mainly phenotypically as a male, but they will have conditions such as sparse facial hair. They won't really be able to grow a beard. They have reduced muscle tone and inability to produce the muscle. They have prominent breasts. So not that this person is obese and has generally larger, it is actually the development of breast tissue, not generally as the size of a normal female, I say normal in inverted commas, would have, but it is breast tissue that is actually being developed. And they will have generally maldeveloped or smaller testes and general male reproductive structures at the same time. So that is Klinefelter syndrome. So those are the 2 that you need to know. You need to know Down syndrome, extra chromosome 21, and Klinefelter syndrome, where it's XXY. So now how are you going to actually have to be able to identify these? Well, that comes to the last application related to meiosis, which is understanding what a karyotype is. So what is a karyotype? This is a karyotype. So it's basically a picture. And what are you looking at here? You are looking at the actual chromosomes of an individual. This is the condensed form of the chromatin network. Essentially what's happening is this individual, this cell, was going through meiosis, and the pause button was put onto it with some chemicals, and then we were able to take pictures of these. So a karyotype is a visual representation or a picture of the chromosomal composition of an individual. So I'm basically a picture of your chromosomes. That's really what we're looking at. Remember, make sure that you know the difference between what the karyotype definition is and what the definition is of a DNA profile. Remember DNA profiling, I take the junk DNA, inverted commas, the non-coding, highly variable DNA to use for comparisons. So, looking again, this is my karyotype picture. So in this particular picture, the chromosomes, the DNA has not duplicated. So this is not the characteristic X structure, but you are looking at 46 chromosomes here. And what has been happening is they have been grouped together based on their homologous pairs. So although they might be like slightly curved and whatever, you're never going to have to actually put these pictures together. You will be given a picture. So you are looking in the non-DNA replicated karyotype of an individual here. What information can you tell from this karyotype? Well, thankfully for you guys, it actually isn't much that you can actually tell from here. Two important things. So number one, you can tell whether the individual is a male or a female. And that goes here where we look down at the gonosome concentrate or the composition, the X and Y composition. So in this particular picture, it just gives you X and you see 2 chromosomes. Sometimes they will put the letter Y here or they won't. Obviously, if there is a Y chromosome, they have to put it there. So in this picture, you are looking at a female. So because there's 2 X chromosomes. If they had a Y, the letter Y here, and there was just a space, and you saw 2 X chromosomes here, the longer ones, then it would still be a female. Don't get confused, because if they put the letter Y here, doesn't mean that there's a Y chromosome. It's just the space that they would use to put the Y chromosome. So that's the first thing you can tell from this. Is the individual male or female? The second thing you can tell is, does this individual have one of the 2 disorders that you need to know from memory? Again, what are those? Down syndrome and extra chromosome 21, or Klinefelter's having XXY. So if we look at this individual, look at chromosome 21. Do they have Down syndrome? No, because they have 2 chromosome 21s. That's what's normally supposed to be there for an individual. Does the person have Klinefelter's? No, because they just have XX. There's no XXY. And that is it that you can tell from a karyotype. I cannot tell from here if there's any other genetic disorders, particularly from there, because it's just the picture of the chromosomes. The only other way that you will be able to tell if there's a chromosomal disorder is if you are given it as a case study, as with all these things. So yes, I could have an extra chromosome 3, for example, or an extra chromosome 10, but you do not need to worry about those particular disorders to know from memory. It will always be given to you as a case study. So let's say, for example, there's something like Fabry's disease or something like that, which I think, as far as I remember, is an extra chromosome 25, or it's another one, or there's one where you get an extra chromosome 5, and they will give you a karyotype and say, does this person have this disorder? And you will be able to say yes or no. Let's have another practice. Here's another karyotype. So in this karyotype, now we see the characteristic X. So in this karyotype, the DNA has been duplicated because we get the X shape. But I still have 46 chromosomes. Remember, you count by the number of centromeres. How many chromatids are in this picture? 92. So what can I tell from this karyotype? Well, first of all, is it a male or a female? Well, I have 1 X chromosome in the space for X, and I have a Y chromosome in the space of Y. So therefore, this individual is male. So note the difference between the X and the Y chromosome. Your X's will look exactly the same, much like the X shape, but the Y almost looks like a half of an X chromosome. That is what a Y chromosome is normally supposed to look like. So this is what a normal male sex chromosome composition will look like. Then, let's look, does they have one of the two disorders? Have they got Klinefelters? No, because it's one X and one Y, not two X's and one Y. Look at chromosome 21. Does this person have Down syndrome? Yes, because there is 1, 2, 3 chromosome 21s. So this individual is a male who has Down syndrome. So those are the two things that we can tell from these karyotypes. So that's really it for karyotypes. Last thing I want to discuss is some AIM-3 considerations for meiosis. So again, these are conversations that you need to have with yourself, not necessarily things that you need to know as an off by heart, but you can get questions and you need to have an understanding of them. So the first one is, what is male and female? So really in the last sort of couple of years, there's been this real surge of debate surrounding the link between sex and gender. So the whole idea being that sex is a biological determinant. What is your genes, XXY? What genitals do you have? Versus gender being something that has become a lot more malleable, a lot more fluid, something we can change, something that is maybe societally driven. You know, is it one of those things where we only deal with something from genetics and then we have the gender being a social construction? A lot of the sort of debate is now coming between, what if you have someone who's intersex, someone who does not have properly formed genitals, someone who has something like a Klinefelter's, which is not a definitive male or female. So I know that the essay from 2 years ago, or the 2018 one, depending on when you're watching this, was that transgenderism has a genetic basis. So this is a real sort of in-depth sort of thing that you can get asked, and there's lots of debate around this. Again, you need to be able to argue both sides of a particular argument. Don't feel that you need to stick to one side. Then, attitudes towards those who have genetic abnormalities. So, for example, if you look at someone with Klinefelter's syndrome, do we accept that person as male or female? Do we accept that person in general society? Do we sideline them? Do we stigmatize them? What are these kind of things? If we think about Down syndrome, yes, it's become a lot more publicly aware. Do we still think that they can be functional in society? Do we still try to awareness campaigns? Do we, is it something that should be encouraged? If we know that someone is going to potentially have Down syndrome, are we going to encourage that? That's really something that is for us to consider, these attitudes towards the abnormalities. And the last one is understanding cause and effect, diagnosis and prognosis, what all these words mean. So we've really kind of heard of cause and effect. We know that we have a cause and will then give an effect. So let's say, for example, the cause is that I have a non-disjunction, which gives me an extra chromosome 21. What are the effects then? I'm going to have the different physical and mental abnormalities that are going to occur. What is diagnosis? Diagnosis is when a doctor or someone who's a professional in whatever industry it is literally going to tell you, this is what it is. Diagnosis is telling you what is wrong with you, or telling you what the condition is. Again, PC-ness, if you're saying that's wrong, people can still be normal. Normal is inverted commas, etc. kind of thing. So diagnosis is what the condition is. Prognosis is, how is this disorder, how is this condition, how is your disease going to develop? And it could range from something as severe as, this is how long you have to live, versus this is how something is going to get worse, this is how something is going to change. It could also be to the positive side. So, for example, the prognosis for someone with diabetes, for type 2 diabetes, for getting better, is pretty good, as long as you do the interventions that are required. So, prognosis, the how and what is going to happen. Diagnosis, what is the condition. Yeah, so that pretty much ends off my main section of meiosis, really. I'm going to do one more video where I go a little bit more applied questions, where we go and sort of just, like I did with the DNA, practice the questions, I'll upload stuff for you, and then we can really get into understanding what sort of questions will get asked on this meiosis. So I hope you enjoyed going through the different sections of meiosis. Please make sure you go through everything. It is a complicated section. Please make sure you go through it in depth because the questions can be quite confusing towards you. So keep up the good studying, guys. I hope you're enjoying, and see you later.